Hi, welcome back to Discovering Patterns in the Microbiome. In this video, we will discuss PICRUSt and predicting functions from marker gene sequencing. When we study the microbiome, we're often interested in either the taxa, so who's there, or the biological functions, what are they doing? And of course, one of the main findings in the Human Microbiome Project was that the taxonomic composition of people's guts varied widely from person to person. So you could have some people with almost no Bacteroidetes and some people with almost 100% Bacteroidetes. And then when we looked at the biological functions, those tended to be much more redundant from person to person. Although for mechanistic explanations, it seems logical that we're going to be more interested in the biological functions. Those are going to get us closer to what's actually happening. So if you have, say, a clinical data set and you want to understand what's the difference between the bugs, say, in healthy subjects or subjects with Crohn's disease, you might want the functional annotations so that you can see that the bugs are for example, producing more hydrogen sulfide. The challenge is that the taxonomic annotation you can get with 16S sequencing or amplicon sequencing, which is now $20 or $30. And then the biological functions require you to sequence much more deeply, so you have to do whole genome shotgun sequencing. We're no longer just amplifying part of the ribosome as a marker, but we're just chopping up all the DNA and sequencing it. So that costs more like $300 or $400. And the question of this video is whether or not we can use this, so the taxonomic profile, to predict the functional repertoire. And we're going to be doing that using this tool called PICRUSt. So PICRUSt provides somewhere between 80 and 85% accuracy for predicting the full metagenomic repertoire in the human gut. And the way it does this is has a number of steps, and so I'm going to break it down. In the first step, we take a tree of all of the known taxa and all of the taxa that are observed in your sample. For some of these, so here, this is the phylogenetic tree. The dark blue circles are the ones, the taxa where we know their genomic content. So we have a strain sequenced here. Then the light gray or the light beige are the ones that, for whom we don't have good genome level functional annotation. So what we do is we first remove the taxa from the tree that don't have their genomes sequenced. So now we've got a pruned tree with only, for every tip we have a full genome, we know the whole genome content. Then we use some simple methods to infer the ancestral states for all of those tips. And we do that for every gene independently. So we know basically what's the count of a particular type of gene in every single tip on the tree, and then we infer the counts in the ancestral states. Then, knowing the ancestral states, we predict back out to the tips for those tips where we didn't actually have the full annotation. And when we do that, we can use standard methods for doing that prediction and also estimating the confidence in the gene counts that we're predicting. And we can do this either for gene counts, or we can just do it for presence absence of a gene. So now we have the full genome content predicted for every species or every OTU. These are all the known OTUs plus any that we saw in your sample. And from that, we can get the gene content predictions for all of those bugs. Pretty straightforward. We can also use this to estimate the copy number of the marker gene, in this case 16S. So we can predict how many 16S copies each bug has in your sample. And this lets us better understand the actual counts of each of the bugs observed in the sample, so that we're not double counting bugs that have 2 16S copies or triple counting bugs that have 3 16S copies. So that's something you get kind of for free in PICRUSt when you're predicting all of the genes. One of the genes is the 16S. Then you combine, so that's all the inference, and that's all done ahead of time. And then when you download the package, you get all of those inferences. And in practice, we're not actually predicting new functional traits for new OTUs every time we do this. To make it run much faster, what we do is we take the whole green genes set of OTUs at 97% and predict those in the tree ahead of time. So that comes with PICRUSt. Then you don't have to wait for it to do all the ancestral state reconstruction and prediction while you're running it. So then you take your OTU table, which has to be, at least for the published version of PICRUSt, has to be closed reference OTUs picked against the Green Genes 2013 reference set. Although PICRUSt can easily be set up to run on other databases. And then you take that OTU table, your OTU table, and the 16S gene counts and run it through a script to normalize the copy number. So that's where you're correcting for copy number. Now you get a normalized OTU table. Then you take that normalized OTU table and the predicted gene count for every OTU. It's basically a matrix multiplication. And that gives you your inferred metagenome. And that's the final result, is basically this table that says for every sample, we know what's the functional repertoire. And the nice thing about this is that we can take the $30 data, that was the marker gene sequencing, and from it predict the full functional repertoire for around, for free, we're getting around 85% accuracy. Here's a picture of the accuracy of PICRUSt on the Human Microbiome Project data, and this is showing just broad trends in the differences between different environments. But still, you can see that the PICRUSt samples are shown with triangles. You can see, for example, the skin samples in PICRUSt are clustering with the skin samples in the actual deep shotgun data. The stool samples in PICRUSt cluster with the actual stool samples, and so on. This is a very informative plot from the paper. It shows the accuracy of PICRUSt predicting the full metagenome. This is using Spearman correlation with on the y-axis, and then the average distance within a given sample of each OTU to the nearest neighbor in the tree of known taxa. And then these are colored by different environments. So of course, the mammal gut or even human gut samples tend to have a lot more known taxa that are near neighbors to the taxa that are in the samples, because that's what we've spent a lot of our sequencing money on. And so that means that the black samples, for example, those are the human samples, they have low distance to the nearest reference genome for the average bug, and they also have higher accuracy in PICRUSt. This means that ballpark for the human gut, you can get 85% accuracy or so. As you move into other soil samples or environmental samples, the accuracy goes down a bit. When you have really unknown environments like these hypersaline environments, these also are a bit shallower, then you don't get very good predictions. And at a certain point, the predictions get pretty bad, and so I wouldn't even recommend using it. But for the human gut, it's not bad, and you can actually find interesting, relevant functional signals in the data just going from the 16S data to the predicted functional repertoire. Here's another plot just showing the accuracy on a per genome basis, plotted against the distance of a genome to its nearest neighbor in the reference database. And so you see, as you get farther and farther away from the reference database, the accuracy decreases, which makes sense. So in summary, shotgun metagenomics can describe the full functional repertoire of the microbiome, but it's expensive. Still costs a few $100s because you have to sequence deeply. PICRUSt can produce much more accurate metagenomes, somewhat accurate metagenomes of 80 to 85% for the human gut from 16S data sets. There are times when you might not be able to do shotgun sequencing, like let's say you have biopsies, intestinal biopsies, and there's too much host DNA in there to get good deep shotgun sequencing. So there could be cases where you're actually going to get better data by amplifying the 16S gene and then mapping that over to the functional repertoire with PICRUSt. You can also use this to go back and mine published data where there isn't shotgun sequencing. Another common approach is to run 16S sequencing on all of your samples, run PICRUSt on them, and then use the PICRUSt results to choose a subset that you want to spend the money on doing the deep shotgun sequencing for. I would recommend that when you're writing up PICRUSt results, you describe them as being suggestive only, because this helps people understand that you don't think that you actually know what genes are there. You know that they're just predictions. So I'm always careful to just have a little disclaimer there whenever I'm reporting the results. And as I mentioned, they're mostly useful on human gut or mouse gut, and especially in the mammalian samples too. So that's all for this video. See you next time.